Welcome again. We were just talking about the very important subject of angiogenesis and the relation between stem cells, in particular small mobile stem cells, and angiogenesis. So again, we have with us Dr. Roger Schechter. He's a specialist in wound care. And you were saying, Dr. Schechter, that the gradient, the chemical gradient within the wound or the necrotic tissue is very important for guiding cells, migration, and the restoration of the tissue, maybe especially the early phases of angiogenesis. Can you elaborate on that, please?
This is a hypothesis. It's not proven, but in general, it's thought that endothelial cells are attracted into an area of hypoxia by some type of chemotactic signal. The signal was thought or posited to be nitric oxide because there's a higher concentration of nitric oxide in the center of a hypoxic wound than on the periphery. Nevertheless, the source of the nitric oxide is in question. Why is that produced? What produces it? Clearly, if you have necrotic tissue with dead cells, they're not spontaneously going to produce a chemical species like nitric oxide. So, is it possible that small mobile stem cells also coming into an area of damage, possibly attracted by hypoxia, could be the source of the nitric oxide that brings endothelial cells in to start the process of angiogenesis?
Yeah, most of our experimentation was based on the premise that the small mobile stem cells will be the one that are the mobile one, and the larger cells will be the less mobile one.
Yes.
So we mostly looked for attraction that is based on the small mobile stem cells moving towards the larger cells, because we hypothesized that such a cell with such high mobility, extraordinary mobility, I mean, you can see the cells moving, that these type of cells are actually more likely to be the invader and the one that is forthcoming with the movement. Now, whether the SMS cells have also the ability to attract other cells, I think that's an interesting concept. It needs also to be tested and checked for. The interaction between specific cells, such as the small mobile stem cells and endothelial cells, are proven to be extremely strong. We have seen strong interaction. And not only that, we have seen also the angiogenic effect of small mobile stem cells, that is mainly the extracellular matrix that is derived from the small mobile stem cells. So it's also consistent with a narrative that these cells are involved in repair. I guess it's obvious that any tissue requires for repair early blood supply, and anything that encourages endothelial cells to form capillaries would be conducive to such a repair.
Interesting. Can you contrast the angiogenic effects of small mobile stem cells and perhaps their extracellular matrix versus mesenchymal stem cells?
Yes, we know actually already that mesenchymal stem cells have an effect in inducing the formation and the tubulogenic formation within endothelial cells. We know that mesenchymal stem cells excrete specific factors that is conducive to the formation of microtubule. This is that can be observed in a standard assay where in vitro you apply the cells to endothelial cells, and you do observe the reorganization of endothelial cells into small capillary. What we found with the SMS cells was actually more surprising, because not only the extracellular matrix in absence of the cells can initiate such an event of creating microvessels, inducing the large morphological changes of endothelial cells. We observed also the self-organization and of the endothelial cells into larger vessels. And these larger vessels are quite extraordinary. We can't see them forming with any other type of cell in vitro, and the larger vessels are large enough for us to see them actually with the naked eye. So we believe that SMS cells can provide extraordinary angiogenic effect that might not be available from other cells.
So given that mesenchymal stem cells don't travel much in their natural state and are present in more solid tissues than in the circulation, is it possible that although they may be present again in the periphery, if tissue is necrotic, one would expect that there would not be living mesenchymal stem cells, because likely they too require some nutrients and blood supply in order to function. And as we've discussed, they're not the most robust cells in the world.
Yep.
So is it possible that there is an interaction with the SMS on the periphery of the injured area with living mesenchymal stem cells, and somehow they act in concert to bring about organized angiogenesis? And from my standpoint in wound healing, the process of granulation, which is the development of an organized capillary bed in an area of injury.
Yeah, I think that's very likely, and we tested the interactions between the SMS cells and the mesenchymal stem cells in vitro, and what we have observed is obviously there was a very strong interaction, a very strong bonding, but at the same time we saw functionally the mesenchymal stem cells enhanced proliferation. And even though we had ideal conditions for the proliferation of mesenchymal stem cells, yet the presence of SMS cells enhanced that proliferation even further. So we suspect that an interaction between the two cells is happening in vivo, and that this interaction will replenish some of the mesenchymal stem cells that actually perished during the necrotic stress situation.
So unlike in vitro, is it possible that the interaction can lead to normal as opposed to degraded mesenchymal stem cells, given that they are proliferating or in effect dividing in order to repopulate the damaged area?
Yeah, there are other consequences of this interaction that we probably are not quite aware of. So far, it is, I think, an intelligent guess to assume that that a strong binding, the one that we observe, which is selective. So SMS cells will not bind to any cell. It will bind to specific cells. And one of them are these mesenchymal stem cells. And we suspect that there could be different consequences of that interaction that warrant observation and examination.
Interesting.
We also think that there is a case for endogenous activation. So in principle, one could build a cell therapy that is based on injecting SMS cells instead of the easily perishable mesenchymal stem cells. And such kind of injection within the blood that is most likely well tolerated could activate endogenous mesenchymal stem cells and have an added effect in addition to the effect related to SMS cells. We would have the effect that is derived from increased proliferation and replenishment of the mesenchymal stem cells within the tissues.
So, on that basis, what other cells that are involved in cellular repair are enhanced or acted upon by small mobile stem cells?
Yeah, that's a very important question that will actually send us to COVID-19, and we understand how important this discussion is, and we would like to reserve a next episode where we discuss how can small mobile stem cells influence cells that are related to the lung regeneration, and how that could help with the regeneration of these compromised organs during SARS-CoV-2 attack. So let's do that next time, and thank you very much for Dr. Schechter for bringing up these interesting subjects.
A pleasure, Dr. Rahmo.